What up, Bio, and welcome to our next lecture on DNA and the replication of DNA or DNA synthesis. I start by talking about mitosis and DNA synthesis because every time a cell needs to reproduce or make a copy of itself, maybe it was damaged, maybe it's a growing organism, maybe it's just skin that needs to replace itself all the time to stay healthy, or the lining of the intestines. New cells have to be made. This is a major process. Unfortunately, mistakes in this process is where cancer arises because DNA is the code for life. It tells us how to make proteins, and any mistake that happens in this DNA replication process can lead to cancer. So if we really want to understand the human body and some of the things that are causing us the most trouble as humans and causing some of the most death, starting to understand replication is kind of important. So today, we kind of get into the basics of it. If you ever go further into biology, we can really get into some of the more detailed minutiae of it and really kind of see how replication and understanding replication can really help us treat cancers on a molecular level and maybe save a lot of lives. And people are doing it right now. So, but there's still a ton to be understood and known. It's a pretty complicated process. We're going to go through it in about a half an hour. I recommend pausing, going back if you don't understand things, and write down any questions you have so you can ask me in person. Okay, first thing I want to talk about is the fact that DNA is semi-conservative. So just know that semi-conservative replication, meaning that when one side is copied, the other side is always the original. So DNA is actually going to get split. It's going to get opened up by my favorite enzyme, helicase. And when it opens up the DNA, that means one side is going to be copied. It is a template, and the other side's a copy. We now have two new strands of DNA, and each one has one piece from the original and should be the exact same. Amazingly, the mechanisms, the enzymes, and everything that run DNA replication, the chances for a error in just one of these little bases, an A, T, C, or G down here, the chances of that are it's like 1 in 10 million or more is the only time it makes a mistake. That's quite remarkable. It's a very, very efficient process. There's actually enzymes that'll come in and edit in case there's a mistake. We have other enzymes we want to talk about that we found that actually go in and can see where mistakes are and stuff. So it is amazing. You're constantly making new cells, constantly making billions and billions of new base pairs, and amazingly, you don't make mistakes very often. Although the older you get, there's still some mistakes, and we start to wrinkle, we start to get all kinds of problems there. So, DNA replication, what is it? I think we've already talked about that a little bit. Anytime I ask a question, I want you guys to think about it too, though. And I'll give you a second. For replication, it is just making a new copy of DNA. And I asked why replicate it, and I kind of already gave us this away. In what process? Well, if we need to make a new cell, we need to copy our DNA. If we need to make an egg or a sperm, you also need to copy your DNA, although some weird stuff happens in there to jumble up the DNA. But that is it. We're going to go through the enzymes of DNA replication. There's quite a few of them. There's actually a lot more than these, but these are the ones that you need to know. I expect you to understand and know so we can start gaining a process of this. And really the reason you have to learn this is just to prove that you can learn complicated steps. And it's, like I said, it can help with cancer too. So first one we're going to talk about is helicase. What helicase is going to do is it's going to break the hydrogen bonds. Right here in my background, you can see the DNA, my little laser pointer is going over it, and holding these nucleotides together, as we talked about in the last lecture, is hydrogen bonds. And we need to open up the DNA to copy it. We need to separate these two so we can put two new strands in, and helicase is the one that does that. If you want a good biology joke, it's a little inappropriate, so don't share this with anybody else. But you're at a college party next year and you want to impress some of your people you're interested in and maybe want to, form a relationship, and you can tell this really great helicase joke, and you can be like, hey, let's make like helicase and let's open up those genes and get inside. Really bad joke. Please do not. Yeah, it's kind of funny though. And you'll remember helicase. It opens up the genes and gets inside. So that is the job of helicase. It opens up the DNA to allow for transcription. So pretty awesome one. DNA gyrase, sometimes it's called topoisomerase as well, but it's going to prevent supercoiling. So when we open up this DNA, it's all wound up tight. We need to make sure that the area that's not wound up as we open it doesn't cause it to spring and wind up even more. So gyrase is really important. It prevents supercoiling, or like the DNA from like being like a spring that just wraps around itself. I think you guys have seen that. When we open up the DNA, it's no longer double-stranded, and to prevent nucleotides from changing, especially thymine, which likes to reverse into uracil like it is in RNA, these single-stranded binding proteins will lay on top of it. We're not going to talk about those much, but I want you to know they exist. So they'll lay on top of each piece. We'll go over that to keep the DNA stable. Enzyme called primase is actually going to lay down RNA on top of the DNA. We're going to talk about why that is. That happens on something called the lagging strand because weird things happened. DNA polymerase 3, and there's a DNA polymerase 1. We're not going to worry about 2, but DNA polymerase 3 is going to synthesize a new strand of DNA by adding onto the primer. So it's going to attach to this primer of RNA, and it's going to lay down nucleotides corresponding to the other side. So it's an enzyme that comes and basically like is laying concrete, or like when you see pavers on the road, it's basically the paver of DNA, and it grabs little nucleotides from the cytoplasm and sticks them onto the DNA, or actually from the nucleus. DNA polymerase 1 is actually going to replace the RNA primers. This will make more sense as we go through it step by step, but I just want you to write these down and have them.
Good morning. Julie Cassidy, Andrew Morris, and Gillette DeVille. Please see your hand. Thank you.
Yeah, those teachers are screwed. DNA ligase is now going to join something called Okazaki fragments. So this will be another vocab word that we come into. And they're really fun to say, Okazaki. Like Okazaki, Okazaki. I like Okazaki. That's the only way I can remember them, is because it's a fun word to say. It's actually named after the scientist who discovered this process. So he's pretty awesome. Okay, so make sure you get those vocab words written down. Kind of try to memorize what they do. It'll make more sense when you see the whole process happening, but I think it helps to have your idea of, okay, when's that thing going to come into play? So I had you guys write those down. Next thing we're going to talk about is 3' to 5'. This is a big deal, and you're probably wondering, what in the hell is 3' and 5'? So this is a diagram of the wonderful ribose or deoxyribose sugar that you find in DNA. And although you guys aren't in chemistry or organic chemistry, when we do have these molecules, we try to name different sections of them. You might recall triose phosphate that we talked about, and that's because things were on the 3rd carbon. So we name them by the carbons in the carbon chain. So this is 1', the first carbon, 2', 3', 4', 5'. So when we make DNA, here's our sugar right here, and this is our phosphate. This is that backbone ladder. You will notice that the phosphate hooks up to the 3' end of one and the 5' end of another. 3' end of one, 5' end of another. And then on the other side, it's the same way, 5', 3', 5', 3'. Well, all the enzymes in DNA run in the 5' to 3' direction. So when we're doing any work, whether it's making mRNA, or which we'll talk about later, or DNA synthesis, it's going to run in this direction. So it will run from 5' to 3', and on this side it runs from 5' to 3'. So these are anti-parallel. They go the opposite directions, and this is going to kind of cause some issues for replication, and therefore we have to know the direction that it runs 5' to 3'. It also helps if you're reading genes, you need to know that they run in a 5' to 3' direction. So here's how DNA replication works. This is why it's a big issue of 5' to 3' too. DNA polymerase 3, so that's the enzyme that's going to add nucleotides to the corresponding original. So we're down here. Here's my original. This DNA polymerase 3 is kind of traveling like this, and the whole time it's adding new nucleotides in front of it, okay? Adding new nucleotides. Well, it has to add to the 3' end, and it must go in the 5' to 3' direction. The issue this causes is when you look at this way, so here's 5'. Oh, did I get that backwards? Oh shit, 3' to 5'. No, it's adding, so this side is 5', right? This is 3', okay? Which means the 3' end is right here. So here's the 3' end. DNA polymerase can add to this 3' end, no problem. DNA polymerase can travel that direction, no problem. But when we look on this other strand, now, if we were to start back here where it must start, which we're actually up in here, and it's heading this way, we have a problem because now this is the 3' end and this is the 5' end. Okay, which means we'd be trying to add on to the 5' end, and it cannot do that. So where this side we get to say is continuous because it's nice and easy, it has that 3' end to add to, meaning we go from 5' to 3'. On this side, we don't get that option anymore. Things are wrong, we can't go this direction, so we still have to grow this direction, but we have nothing to attach to. So that's where this primase comes in. We get to lay down some RNA, and by putting that RNA down, which can be laid down on nothing, we now have a nice 3' end. So this would be a 3' end to add to, and we can actually start building it. But this causes issues, so we have to do this in sections or fragments called Okazaki fragments. And we'll go through that again here in a minute. So the leading strand, more vocabulary, this would be the leading strand on this one, okay, leading. Easy continuous replication, it goes from 5' to 3' direction. This is going to be like, you're going to be like, wow, I understand replication, it's easy. DNA polymerase 3 comes and just adds nucleotides in the 5' to 3' direction. The lagging strand, unfortunately, is in the 3' to 5' direction from the origin of replication. That's where it starts, not too big. And it's going to get really fancy. So it's going to get more difficult, and it brings us to our next vocab word, the Okazaki fragments. And all this is DNA is replicated in small fragments on that lagging strand. Okay. Ligase is also going to be a new enzyme, and it's used for joining these fragments together. Okay, so easy in the leading strand. Everything's 5' to 3' direction. We just need DNA polymerase 3 and nucleotides. We can copy that DNA really easy. The lagging strand, because DNA polymerase has to hook onto that 3' end, it gets more complicated. We're going to have to lay down some RNA to give a template to start. We're going to have to do it in multiple sections, and then we're eventually going to have to join these small little sections together, and that is ligase. So let's now actually go through the steps to DNA replication. We'll start with the beginnings, then we'll do the leading strand, followed by the lagging strand. So starts at the ORI. This is the origin of replication. This is for bacteria. It's for prokaryotes, not eukaryotes. We have an origin of replication as humans too, or eukaryotic cells. But remember, DNA is circular on a bacteria, so it's a little easier. On us, it's not, and we have like millions of different origins of replication in our DNA. So we don't need to study that. It is much more complicated, and I'm happy we don't. Okay. By the way, it's semi-conservative. We talked about that. So first thing that happens, helicase is going to open up the DNA. So right here on both sides, well, actually it would have been right here in the center, helicase opens everything up. That gyrase right here in green is going to hold this piece together to keep this stuff from doing crazy stuff and supercoiling. And on each end of the fork, so here and here, picture that DNA is opened up, we're going to get a replication fork. So this is what we call the replication fork, where it's splitting. Okay, that would be happening somewhere over here too, but I don't have any diagrams that show that, and it would be kind of complicated. Okay, then on the leading strand, really easy, way back here, you'd have your replication fork. DNA polymerase is just going to start running along here, going this direction in the 5' to 3' direction, and it's grabbing little nucleotides that correspond to the side. Remember, they have a bunch of phosphates on them, so the energy to do this comes from them, and it just nice and easily lays down new nucleotides and makes a nice new copy for this to go be a new piece of DNA in the new cell. So DNA polymerase 3 adds nucleotides in the 5' to 3' direction. We're going to keep saying that, 5' to 3', and that's because it has to add on that 3' end right here of the other nucleotide. This continues, and like I said, it's fairly simple to remember. I mean, that's pretty awesome. We just lay down, DNA polymerase 3 just comes and adds nucleotides. Now we get to the more complicated part. Okay, replication on that lagging strand, like I said, it's got fun words, but not as fun to learn. Okay. Before I go back with the hard part, I do want to talk about when we'd have these open DNA pieces and it's single-stranded. That is where those single-stranded proteins, there's little proteins that sit on here to keep these from interacting with anything else. It's not pictured, but as polymerase comes through, those pop off as the new nucleotide comes in. So those binding proteins, that's what they're for, is to prevent these from doing anything else, and they'd be all over it. Okay, so the fun word, Okazaki fragments. So remember, these are going to be little tiny fragments on the lagging strand where we're going to start DNA. So here's kind of a drawing. There's a little fragment, Okazaki. There'll be another one here, so that little short stranded ones. And this is all happening like simultaneously. So this one's growing at the same time as this one, at the same time as this one, while this one's going completely on. Okay, so I'm going to kind of discuss how this happens first, and then we'll come back and take notes. Let's see if I have a big one. So, real quick review on the leading strand. DNA polymerase is just going to come in and lay down new nucleotides corresponding. So if this is A, this is T. If this is C, this is G. And it's just going to lay those down in the corresponding direction. Okay. This is after helicase has opened it up and our gyrase is keeping everything held together. On the lagging strand, because it's going the opposite direction, okay, things get a little weird. So remember, this one started up here. This one's going to start all along here, and the first thing that's going to come in is our primase is going to come down, lay down little pieces of RNA, and it's pictured in red here. And so all along this strand, you have multiple primase enzymes starting this little, maybe it's 5 to 10 nucleotides of RNA. Okay, I got to finish this up quick. I'm behind. Good morning, baby. Okay, so RNA primase is going to lay down the RNA, and that's going to give a template for DNA polymerase 3 to come and start adding nucleotides going in the 5' to 3' direction. So all along here, they keep building these, and they happen at the same time, but you get all these little fragments. Eventually, the RNA polymerase 3 will run into the RNA of the Okazaki fragment in front of it, and it'll stop, it'll release, and we end up getting like a red RNA and then blue DNA, red RNA, blue DNA. That's where DNA polymerase 1 comes in, and it's going to write over the RNA and turn it into DNA. And then when we have little gaps where this has formed, DNA ligase comes and fuses them together, and that completes protein DNA synthesis. I don't know what just happened. What the? Okay, sorry about that, guys. I deleted my PowerPoint.
Hey, nerds, what are you doing here? Get alive. Sit down.
I didn't finish making my video.
What?
Get out of here.
How are you gonna do it?
And then these kids come and fucking bug me early. I should have locked my door. But somebody came in and unlocked it.
Who's that?
Especially that one I don't want in here.
Yeah.
Oh my gosh, I just said that on my pre-recorded video. What? Okay, so senior bio kids, I'm going to finish this up real quick. We kind of went over it. I was late and class is starting, so I'm going to finish this up quick. So here's how on the lagging strand works. Okazaki fragments are started. Remember, primase lays down the short strand that gives us somewhere for the DNA polymerase to start on. Then the DNA polymerase works kind of like it does on the leading strand and adds them till it runs into that RNA strand before. Then DNA polymerase 1 is going to replace the RNA with DNA. And we now have little gaps, and those gaps are going to be filled by ligase. Okay, that is it. I'm going to leave you guys with just this last part that goes through everything. You can read that. It is in the PowerPoint. I recommend you look at it. Have a wonderful day. Enjoy your questions today because I don't think you're going to have